All right. Hello. Welcome to this webinar on antibiotic discovery and development with LabKey's Biologics LIMS. Thank you very much for joining. Really appreciate some of your time and attention today. My name is Bernie Lee. I'm a product manager here at LabKey, and I have a short presentation for you. We'll be briefly talking about some of the data management challenges in biologics discovery and development, and some of the tools and strategies we employ here to drive success, improve efficiency, and increase quality of life for scientists, and ultimately what that facilitates. I want to spend most of today in the product itself through the lens of our biologics LIMS. I'm of the opinion that that you shouldn't be running, 3 or 4 or 5 or 6 different pieces of software to help manage your data. You don't need an SDMS and a LIMS and an ELN, separate analysis software, and a bunch of spreadsheets in order to do this. There do exist products like our Biologics LIMS that are really as close to all-in-one scientific data platforms as you can get. And I just want to give you a glimpse into what that looks like and what some of the advantages are of using tools like this. So ours, LabKey's Biologics LIMS, was originally designed for antibodies in particular, but we support other kinds of biologics that you might discover, develop, and we have all these myriad components or tools within that we'd like to talk about. Extensive sample management tooling, the ability to capture information about all of the various entities that you may be working with through your scientific process, the ability to characterize all of those different samples through assay data management, a lot of reporting and visualization stuff, built-in ELN, and much more. Those tools facilitate accurate tracking of samples, all as well as the plates in which you will probably primarily be operating in, as well as giving you a centralized place for all of this data that's consistent and allows for finding your data faster, quicker insights, faster decision making, stuff like that. So there's definitely some serious challenges for biotechs designing enzymes, designing or discovering antibodies and building them out. A select few presented here are things like lost or confusing data, people losing track of samples, losing data itself, or an inability to make use of data after the fact because it's stored in a PowerPoint presentation or in some standalone ELN, or in somebody's file system somewhere with a strange label that nobody knows how to interpret besides the scientists that put it there in the first place. There are, relatedly, insufficiencies in post hoc utility and the ability to track back, given that discovery and development often require multiple iterative screening rounds, lots of handoffs between scientists. It can be particularly difficult to go from whatever one downstream scientist is working on back upstream to relevant relevant sequences, clones, or batches of molecules upstream, and the proteins that they represent. And then, of course, something big these days in that post hoc utility space is trying to make use of your structured data or unstructured data for things like predictive algorithms, machine learning algorithms, and other such data science tools that allow you to better design, better predict the success of the molecules that you're actually operating on. And the last thing I'll mention here is that there's a lot of wasted time, lots of time lost when you have to redo experiments or spend a lot of time digging up old data. You have to reconstruct analyses, reconstruct reports, and have lots of meetings with lots of people in order to be sure that you've compiled the right data for whatever the decision is you're trying to make, or whatever the report is you're trying to submit, or for an IND or something like that. And so, by having a centralized system like this, you can better ensure reproducibility across all of this stuff, as well as accessibility. That's probably one of the first benefits that I've listed here. Yeah. So what you really want is a solution that provides, some of the following benefits. I was just talking about that centralized application, where you can, register the entities that you're investigating, and all of the associated data with it. You want that additional data to be properly associated. You want it to be integrated with with the samples that you're dealing with, the plates that you're working on, the assay data that you've generated, and you want that to include the the analyses, the hit selection criteria, the things that you're operating on at a bench level. You want that to be well integrated, probably automatically with with the upstream stuff upon which you are actually trying to decide. You want somewhere to document your operations for improved reproducibility and an understanding, a more objective understanding of what you actually did and why, as well as an easy way to return to your data for insights, decision making, planning future work, stuff like that. And of course, controlled access, full auditing, so that you can actually solve problems that arise from your data. Yeah. And with that, I told you short and sweet. I want to jump over and show you some of our application. So let me switch share here, and I'll show you some Biologics LIMS. Come on, Zoom. There it is. All right. Okay. So I dropped you on this page in LabQ's Biologics LIMS. We are looking at details about Herceptin Fab. Now, Herceptin is a commercial antibody. I chose this one just because it's recognizable. I wanted to start with the way that you can register and capture information about the various entities that are relevant to you. So this is a molecule. It's an antibody, but you can capture information about your cell lines, about various chemicals, various compounds that are part of your process. You can capture information about the vectors that you use for your different purposes, as well as in this case the protein sequences and the nucleotide sequences that make up these molecules. So here I clicked into the heavy chain for this molecule. I wherever or however I've done the sequencing for this, I can then take that information, register it in the system, see information about my frameworks and CDRs and all of that kind of good stuff, as well as understand important physical properties for these sequences. I can go even further and look into the nucleotide sequences that make these up, or back up the chain and examine the molecule itself that we've constructed and then worked on. Those relationships are really important when you are ultimately trying to keep track of everything that you're doing and ultimately make some choices about what molecules are the best fit for your purposes, or what yeah. And so in addition to that, there's some significant connectivity built into this. I can see just at a glance how many batches of this molecule I've actually produced, as well as things like what notebooks this particular molecule has been written about in. So if I come into these protein batches, I can see a whole bunch of samples in this enriched samples grid that shows me a bunch of information about, in this case, the different names of the samples, where they are in storage or whether or not they're in storage. In this case, I've pre-filtered this so that these are non-aliquots. So these are the original samples, their status, information about their volume, what molecule they originate from, what expression system was used. So we can really enrich these data grids so you can understand all of the information about the actual materials that you're working with. And if I click into one of these, I can see, specifically which location and which box these things are, how many aliquots this one might have. But it also gives me the opportunity to show you a lineage view of these entities. So the sample that I'm actually looking at, I can see that it has these, we'll call them parents. I take this dual construct that has a couple of nucleotide sequences inserted into it, combined with a particular cell line to produce these actual batches, which I've harvested and then I can experiment upon. Anyway, we have all of this data connected, and then what that allows you to do is really profile and track down known entities generated throughout whatever actual processes are going on. And so some of those processes might be actually producing these batches of protein and then characterizing them. And one of the things I wanted to show you really quick today is actually taking some of those batches and characterizing them. So in this case, what I want to do is take these and put them on a plate so that I can run some kind of, I don't know, activity assay on it. So this is activity assay or whatever it might actually be. And I have prefabricated a set of plate templates for my various uses. And in this case, I actually want several of these with different configurations. So I will select those, and then, oops. Uh-oh, what did I just do? Sorry, saved the wrong thing there. So we'll just redo that manually. My apologies. We want to get the right set of plate templates in here, and then I'll show you how we automatically distribute those samples and help you make use of these. I'm just going to kind of arbitrarily name these for demo convenience. This is what I was trying to paste in, sorry. And then move on. Okay, so I've taken a basic selection of relevant protein batches, and I've said I want to put them on this plate, on this set of plates. And what happens is we distribute those according to the template. So we have a dilution curve for each of the different samples that I've put on here. You can see that the first one of these and the last one of these are replicates, and the rest of them are the 5 samples that I selected. And I have 4 plates worth of these things that are slightly different, ultimately producing a whole bunch of replicates of the same dilution curve. I didn't have to do any additional work right then in order to get these things to be distributed properly. From here, I can tell the system to generate either a work list or some instrument instructions so that I can just feed these plates directly into my plate reader and ultimately get the data back out and import it. I'm going to step through that really fast. I'm going to import some of this data. You won't see this part, but I'm going to drag a file over. You did see that. And that file is just a multi-plate plate read. And what happens is it imports and it automatically associates the raw read, so whatever value is in that particular well, as seen here, with the sample that I put in there and its location and the particular plate. When that data comes in, not only do we do that association automatically, we also do things like calculate means and standard deviations for you and automatically generate appropriate charts. So in this case, we're doing these, replicated dilution curves to produce a curve fit and figure out where my, for example, EC50 might be. All right. There are a variety of additional analytical components that you can take advantage of in the system once this data is made present. You can mark relevant data as hits, or you can have our system automatically do it. So if in any of the assays that you're running, you want some you want a particular threshold set so that anything above a particular value is marked as hit, you can do so. You can also establish these things manually. We also provide some quality control examinations. If you have major outliers, if this one in particular is a major outlier, I can mark it as such and exclude it for the purpose of these mean and standard deviation calculations, as well as the curve generation itself. Now, so obviously that kind of ruins that particular curve, but that's okay. It's for demonstration purposes only. All right. So what I showed you right here was just a direct assaying, a direct characterizing of existing batches. We also support highly iterative operations, which I will give you a quick glimpse of, but I'm not going to spend a lot of time on. So as you might be able to see here, I have a primary plate that I am extracting material from in order to run, in this case, an ELISA. And as I move through this process, I can examine those particular results on the downstream plates. So what's very common in, say, a phage display discovery campaign is you start with some set of material that you want to screen and work with. You do some of the you do some of the screening, and then ultimately you want to quantify something. You want to use that quantitation in order to make some selections. Here's what some of that automatic hit selection looks like when you import some of this raw plate data. And then from that original material, so not from the assay plate that you're ultimately going to toss out, but from the original material that you extracted it from, you can choose your hits, and you can pick them to go onto a new plate for the next round. And that's what I've done here. I've picked a subset of the original samples and put them onto a new plate for whatever the next operation is. So I really only showed you 1.5 of the supported activities around plates in the system, but again, just supposed to be a quick demo here that we can then circle back on in a private conversation to talk more about your particular use cases. Okay. Oh, yeah. Okay. Talked about exclusions, hit selections, and I think I talked about roll up. Okay. So I showed you how we could capture information about those molecules. I showed you how the protein batches themselves can be used in a lot of those plates, and I think I showed you some of the relationship between all of the various entities. I believe I showed you one of these lineage relationships where you have the components that go in and ultimately produce those batches, and when you run assays on those actual samples, one of the other things that we do is roll that information up for you. So if I go and I actually look at this molecule again, I produced a whole bunch of batches, and some of them I ran assay data on. And so if you want to be able to ask questions of your data without needing a data scientist or statistician to come and help you, we will automatically say, okay, here is all of the data that you have for a particular molecule. I can see some chromatograms that I've captured for this. I can see similarly some page gel images for these. I can see some various absorbances, protein quantitation. kind of information. You can see which samples that information was run on. And if you want to go back and find more of that material to do something else with, it's as simple as clicking into it, and you can see where you have more of that material in the system. Okay, there are a lot of other pieces of the system. I only briefly even mentioned storage. I only briefly mentioned notebooks and the fact that you can, in this data connected fashion, describe everything that you're doing as part of your process. I showed you how you can capture assay data, but what I didn't say is that I've pre-configured a whole bunch of assays in here, but all the different types of assays that you run on your materials can be configured in here as well. Yeah, just a taste. So let me switch back. Where's my window? There it is. Haha. Okay, so my hope today, by just kind of briefly touching on some aspects of Biologics LIMS, was to just give you a taste and invite you to consider having a conversation with me, about your particular use cases and your particular problems, so that I can produce something much more tailored towards what you actually do, so you can see more of the benefits of the system. But just to reiterate some of them, when you have a comprehensive data management solution like we offer, you significantly reduce your dependency upon spreadsheets, which reduces errors. It reduces the time that is required for you to track and report on your various operations. It allows you to ultimately keep track of your materials a lot better, both for reproducibility as well as using that material for something else in the future. We will help you maintain the uniqueness of the entities in which you're operating. That's your molecules, your sequences, things like that. We will really benefit your operations by speeding up a lot of what happens. A lot of the data captured analysis, as you could hopefully see, can be automated. LabQ can simply listen for new data as you produce it, import that data, and form the right association. So you can simply step up, look at your analysis across multiple assay types and things like that, and make decisions about what you want to continue to act on. Again, we have this integrated data connected ELN that's built into the same system in which you're capturing your actual data, so that you can comprehensively document and report your findings. And what you ultimately end up with are fewer regenerated reports, happier scientists, happier managerial staff, much happier data scientists, and ultimately a lot less energy is required for you to be successful in your scientific endeavor. And so with that, just want to offer my thanks one more time for taking this initial tour of LabQ's Biologics LIMS. I would be delighted to meet with you, learn a lot more about your particular use cases, your goals, your pain points, excuse me, and see if we can help solve them for you. At this time, I would, if you would like, answer any questions that you might have that you would like to air publicly. And if not, please get in touch with us, and we can save those for a future discussion. So thanks again, and let me just see if there are any questions out there. Okay. There are. Uh-oh. Someone said they lost audio. Okay. We'll make sure to send you the recording if you would like it, and be happy to settle up a little time to have a conversation about the end of this anyway. And someone asked about LCMS-based assays. Yes. Okay. So mass spec results that would come out of an LCMS. So we have a variety of people that will use for complex assays like chromatography and mass spec. A lot of people will take the summary data conclusions. Oh, sorry. Let me back up. I showed you how you can capture some of that unstructured data, some of the images, things like chromatograms, page gel images, things like that, and have those associated with the samples in which you're operating, and how those are automatically associated with the molecules that you might be characterizing. So that's often one part of those. If you want those mass spec traces captured in the system, you certainly can. As more often than not, what I see is that when you have tons and tons and tons and tons of those traces, people don't necessarily want to see and reanalyze the traces within our system. They want to capture that information, and they want to capture, critical summary information about, the original reason why they were using LCMS in the first place. And so they will capture maybe some of those peaks with calculated information derived from the mass spec analysis software, and associate that with what they have, sometimes with a link over to the original data or application in which it was analyzed. So there are a variety of ways in which people have done that, and I'd be delighted to show you what that kind of looks like, and hear about your purposes therein. Thanks for that question. So thanks again a lot for a little bit of your time today, and look forward to seeing you soon.